Hi, thanks for joining today. This video will cover setting up a bulk RNA-seq experiment in Pluto. We'll be using this example here to really take through end to end. This is a published bulk RNA-seq dataset. You can see the original paper here, and the FASTQ files were posted on Geo, so we were able to download that data and we'll be using it for an example today. This is a liver chip model for alcohol-associated liver disease. Moving back into Pluto here, we can see a preview of what we'll be able to do once the RNA-seq dataset is fully set up. We'll be able to create lots of different kinds of analyses, view QC report for our pipeline, and many more things. So let's get started. From our workspace here, you'll see that I do have a lot of projects and experiments in my workspace already. Yours might be starting from scratch. In any case, I'm going to start by creating a new project just to give us a clean place to start from. I'll save and close that project and we'll see it appear here on my lab space. You can see that there are no experiments in this project quite yet, but all of my team members have been added, so they'll be able to see this new data set. Clicking into the project now, you can see that no experiments are in this project yet because we just created it. So we can go ahead and click Add Experiment to start uploading our data. In this case, the project has already been selected. I could always select a different one if I wanted. In this case, I might have study number 1 for this assessing or characterizing the liver chip model. As a next step, we'll scroll down here and select the organism. These were human cells used in this experiment. I'll be able to select the particular genome build for the organism that I select in a later step. Next, we'll select the pipeline or experiment type. So this will be a bulk RNA-seq dataset. And then we'll click Continue to move forward in the wizard. Now that Pluto knows we're uploading bulk RNA-seq data, it's going to show us a couple of different options here that are based on that experiment type. So we can upload FASTQ files. This is always what we recommend because we can really start with the QC information and make sure that the sequencing was high quality before we proceed with analysis. However, if you have process data, you can also upload that in a CSV format here by selecting the units, such as raw counts, checking out one of our templates in case it's helpful for formatting your count matrix. And then there are other ways that you can import data into Pluto also, such as inputting FTP credentials from a sequencing vendor or connecting one of your existing Google Cloud or AWS buckets. For this example, we'll go ahead and move forward with uploading a FASTQ file. You can also import from BaseSpace or other locations. I just dropped in a single file, but you can also bulk drag in several files at a time and you'll see them all queued down here for upload, one at a time. So we'll take a quick break while these files upload and be right back. All right, so coming back to this window here, we can see that 14 files have now been uploaded. These are FASTQ files that are either R1 or R2. This is a paired end experiment in this case. And the FASTQ files have finished uploading for all of my different samples. We'll move on to the next step and actually associate those FASTQ files with different samples in this study. So to move on from this page, I'm going to say I'm done uploading files. And this will kick us over to the next step in the wizard. On this step, what we'll be doing is annotating our samples. So we can open up a sample data template if we're interested in maybe doing this the first time and learning about it. We can make a copy of that. I'll also just change this to view so we can take a look. So here are our instructions and example template. This really breaks down for you different kinds of column names that you might have in your study. You'll see that the required column names for an RNA-seq study will need a sample ID, and then if we're uploading FASTQ files the way that we just did, we'll have this R1 and R2 FASTQ file column. The rest of the columns that are going to go into your table are really up to you. The only rules here are that they have to start with a letter and then only have underscores in here, so avoid any special characters besides that underscore. Looking at the example sample data on the second tab of our template here, you'll see unique sample IDs in this first column, the R1 and R2 FASTQ file column here, and then some information. And again, this can really be whatever I want and whatever biological variables I had in my study. So if I have genotype or dose or other variables, I can just add those as additional columns. For the study that we were just looking at, so over here on the assay data tab, again, for these studies, I did go ahead and start a sample data table for us, and you can see that here. So these are the file names for the 14 files that I just uploaded. And then I've given those samples a corresponding sample ID and matched up the R1 and R2 FASTQ files here. So for this first vehicle sample, it had paired end sequencing files here. So these purple columns are really the only ones required by Pluto. And then you'll see here in the gray columns some additional information that I've added. But note that I can edit these in terms of order or contents. So for example, if I wanted to include another column here, like replicate, I could do that and include some additional information. So really what you want to do here is just capture as much biological information as you have about the samples in your study. In terms of column order, the only requirement here is that the sample ID stays in the first column and everything else can be ordered however you like. So I actually like to move my R1 and R2 FASTQ file columns here to the end because they'll only be used really during the pipeline, but what I really want to focus on when I'm doing analysis will be these columns here because this is what I'll be doing my comparisons based on. So I can download this file here as a CSV format, and that's what we'll be uploading to Pluto. So going back over here to our sample annotations, I can upload the sample data table, and you'll see that right away Pluto opens it up in this preview window. So importantly here, if I had any issues with my sample data, let's say I had some weird character here or this happened to start with a number, you'll see that Pluto tells me what I need to fix about the sample data. So in this case, sample IDs have to start with a letter. So we want to remove that 1 from the start of the sample ID. But besides that, you can continue to make edits here. So I could add another variable like replicate if I wanted with a default value, say of 1, and you see that that's added the column over here, and then I could edit inline, just double click to change any of those sample annotations. When I'm happy here that everything has matched up correctly and I'm not seeing any red errors, you can see here a nice confirmation of that there are no issues found. I'll go ahead and save and continue. What this screen now is showing me is that I need to double check the conditions that I have in my sample data table before doing analysis. So right here, these will be the different variables that I'll be able to do downstream analysis based on. I can look at tissue, I can look at donor, replicate. This is really just a good reminder that if there was anything else in your sample data or in your experiment that you wanted to capture, you should double check that before moving on to analysis. I'm happy with how this all looks, so I'll go ahead and save and continue. We'll see that the upload will finish for that edited CSV file that I've now uploaded. And this fourth step here has appeared to configure a pipeline. So if I continue on, Pluto knows that I've uploaded FASTQ files, and I've also uploaded some sample data. So in this case, you can see here pretty clearly that I'll have a paired end sequencing experiment because I have 2 FASTQ files for every one of my samples. At this step, we're going to set some basic parameters for running our bulk RNA-seq pipeline. Now, Pluto uses industry standard NF core pipelines that have really robust default parameters that reliably scale and work across a wide variety of experiments. This means that we don't have to worry about selecting too many parameters at this stage. We can really just focus on the scientific questions that we have and how those might affect our choices. So here, for example, I may have a specific genome build that I'm wanting to align against. The newest one is pretty common, but I might be choosing an older genome build if I want to make sure that my data can be compared to an older data set. I'll go ahead and pick this newest genome build here. This is a paired end experiment. We've already covered that part. And then RNA-seq has a parameter called strandedness, which is some kits for RNA-seq that you can buy use forward strandedness or reverse strandedness or unstranded. If you don't know the strandedness of your experiment and the way that that chemistry worked in the kit, it's no problem at all to just select auto detect. It might take a little bit longer, but really it's all pretty hands-off. Now that we're happy with these parameters, we can click Run Pipeline to kick off an auto-scaling bioinformatics pipeline in the cloud. What this pipeline is going to do is take those FASTQ files that we uploaded and perform all of the processing, quality control, trimming, alignment, all of the other steps that are needed to ultimately convert those FASTQ files into a count matrix that we'll use for downstream analysis. I can see here that a pipeline is now active and in progress, and I can refer back to the parameters that I selected. In the meantime, this is now a great time to go to my experiment and add any other information. So you'll see on my overview page here, I have a full notebook where I can add information. I have a basic overview as well. So if I wanted to add a link to this original paper, I can edit that overview and add this custom link, for example, and save that. And now we can see that that link is added here. In my notebook area, I can add header, images, texts, lists, any information about the protocol maybe that was used that might be relevant to anybody else that's viewing this study. Now the pipeline is running, as you can see that this banner indicates, and here as it states, the pipelines may take several hours, especially if you have really large sample sizes, but at this point we really don't need to do any further action on this study. This banner will update here shortly with real-time updates about the pipeline and what step it's at, and every user that's shared on this experiment will get an email notification when that pipeline is completed, and then the data is ready for downstream analysis. So to view any of the users that are on this experiment, I can go up here and open this Manage Sharing menu. I can see the users that have been added to this experiment, and I can also add new users if anybody else needs view or edit access. Another thing that I can see on this page here is the change history for the experiment. So we haven't done a whole lot quite yet, but if I click this icon, or you can also come up to the Collaboration tab, I can see the detailed change history here to see what all I had edited about the experiment and at what time. So here in particular, you can see that I had uploaded the sample data, that I started the pipeline at 6:57 local time for me. And this will help us be able to judge later on when that pipeline completes, how long the pipeline took overall. All right, so at this point, I can head back to the Overview tab and continue editing my experiment, or go out to my workspace and do any other work that I have in progress right now. You can see this experiment that we just kicked off has the current pipeline running. And again, I'll be notified by email when that pipeline is ready and the assay data is ready for downstream analysis. We'll see you in a follow-up video as soon as the pipeline is done running, where we can talk a little bit about how to interpret the QC report and take on next steps.